Hello and good morning to today's NanoMedicine-Austria webinar. I hope you hear and see me well and you enjoy today's webinar. The webinar today is focused around PHOENIX, the Open Innovation Testbed for enabling nanopharmaceutical innovative products, presentation of its open call and the expertise of the Austrian partners. So having a look on the agenda, we start with the introduction of the NanoMedicine-Austria platform. Then it is followed by a presentation of the PHOENIX Open Innovation Testbed. And then we focus around the Austrian partners' expertise with 3 separate presentations. And then before we close the webinar, we have also a Q&A session where any questions will be answered. So here's some technical instructions. If you want to raise any questions, please use the questions section to add any questions from your side. We will ask the questions at the end of the webinar in the Q&A session. You can also click on raise your hand if you want to say something. And also the recordings of the webinar will be made available afterwards. Okay, with this, let's go into the introduction and the presentation of the NanoMedicine-Austria platform. So what is NanoMedicine-Austria? It is a national technology platform addressing scientific and technological challenges in the nanomedicine field. And it brings together bio and nanomedical experts to promote science, research, and translation to the clinics and the market of nanomedicine. So it is really about networking, community building activity to connect the different experts in the fields and together have a broader impact than as a single person. What are the topics addressed in NanoMedicine-Austria? So you see the more or less classic nanomedicine topics ranging from diagnostics, medical imaging, nanotherapeutics, vaccines, but also regenerative medicine. And then we have horizontal topics that address all the different pillars, starting from bioinformatics in nanomedicine, nanospecific characterization, but also target and controlled manufacturing of nanoproducts. And last but not least, also standardization is one key topic that more or less is horizontal around all the other topics. So what are the platform's vision and mission? So the vision is to increase the visibility of nanomedicine as a benchmark for research and innovation at national but also international level. And of course to promote Austria's excellent basic and applied research to overall strengthen the transfer of nanomedical applications from lab to clinical practice. And the specific mission is to build references on Austria's nanomedical research and innovation capability and to achieve a joint impact through community-driven activities. So it's really a platform that needs active members and active experts and players in this field. And then we try to make an impact all together. About the specific objectives of the platform. So of course, one objective is to tackle medical challenges to achieve a more efficient and affordable healthcare, as well as new solutions to improve medical treatments. Then another objective is to contribute to a better understanding of biological processes in the human body at molecular and nanometric level. Then, of course, very important because it's a platform and it's about networking to connect experts in the nanomedical field. And then also to create synergies through multidisciplinary cooperation. Next to that, from the platform, we try to support the Austrian nanomedical community and also provide this link to the European and global initiatives. Then we aim to enhance access to national and international research projects and funding. Then we also provide support in nanosafety, quality and sustainability issues to meet all the regulatory requirements. And one objective is also, of course, to facilitate the close collaboration with industry to also allow large-scale manufacturing production of efficient and safe nanopharmaceuticals. What are the activities behind? So now I talked about vision, mission, and objectives, but what is really the task behind? So we have regular meetings. The chairs meet on a regular basis. We also have biannual meetings with all platform members, and we also organize annual webinar and networking events, in which webinar you are sitting now. Then one activity is also to refine and update the platform's strategy and to also contribute to relevant position papers. Then also to generate and collect possible topics and ideas and also contribute them to national or international funding agencies. So to make them aware of what are the specific topics that we would like to address also from a research perspective. Then one activity is also to support in the organization of workshops and trainings for students and early stage researchers in different nanomedicine related topics. Then another activity is to support with grant applications. So because we are a platform and a network, we of course know many experts in the field and are happy to connect you also with them. And one activity is also to create outreach to the relevant stakeholders, including also targeted public communication. So we also have a LinkedIn account of NanoMedicine-Austria and try to keep the followers posted there about latest news and activities. So about the structure of the platform, so you see in the middle is the core group, that is the general coordinator together with the chairs that support in all scientific matters. So from the coordination perspective, it's myself, Susanne RESCH from BNN, and then we also have 4 chairs that are dedicated to support the platform. Then we have a board of members that are all active members and that contribute to the platform. And then, of course, we also interact with the wider community, international organizations, other platform clusters and stakeholders. And for the chairs, I'm happy to introduce to you Daniel Baumgarten, Ramona Jeitler, Thomas Mohr, and Sebastian Schwamminger, who are all in the webinar also today. So I give you quickly the floor to introduce yourself and your expertise. Starting with Daniel.
Yeah, good morning also from my side. Daniel Baumgarten is my name. I'm from UME Tirol. And one of our research topics here is magnetic nanomaterials for diagnostic and therapy purposes, also modeling. And in this respect, I'm also active in national and international standardization organizations and be happy to provide this expertise here. Thank you.
Thank you. Ramona?
Hello, good morning, everyone. My name is Ramona Jeitler. I'm senior scientist at the University of Graz. I'm a pharmacist by education, but I'm also affiliated to the RCPE, where Johannes Bauer will be presenting his current project. My field of research mainly covers the preparation, characterization of differently structured nanostructures for nanodrug delivery. But I'm also interested in the continuous manufacturing of such products in a very safe and sustainable way, but also in a targeted way so that we end up with products with defined characteristics, so being safe and efficient. That's our main research area. Thank you.
Perfect. Thank you, Ramona. So I hand over to Thomas.
I'm sorry for the technical difficulties. I'm here with my cell phone because my browser didn't work. Anyway, my name is Thomas Mohr, and I'm associated with the company Science Consult, which I founded, and the medical university as well as the University of Vienna. To put it bluntly, we are more or less the computer nerds in this action. Because my main field is bioinformatics, so to be more precise, the analysis of omics data using network-based methods and partly machine learning and artificial intelligence. So that means if you have some nanoparticle or nanomedicine derived omics data from any experiment, then we are probably the guys to ask to put the biological context into it. So I hope you have a nice webinar, and thank you very much.
Perfect. Thank you, Thomas. So now to Sebastian.
Thank you. Good morning also from my side. I'm from Medical University of Graz in the division of medicinal chemistry. My focus point is I'm an assistant professor in nanomaterials for drug delivery, but my main focus topic is bio-nano interactions. So basically everything what combines nanomaterials and biological materials and the interaction with nanomaterials and cells, but as well different drugs and drug delivery vehicles. So that's it from my side, and I'd like to give back to Susie with the presentation. Thank you.
Perfect. Thank you very much for this presentation of yourself. And also thank you, of course, for being chairs of the platform now since half a year already. Okay, so just to inform you also that, of course, NanoMedicine-Austria is an open platform to which any interested organization or expert can potentially contribute and also participate at any time. Of course, given that you have the needed expertise and interest in the nanomedical field. So you see here the current list of institutions and organizations that are part of the platform. So if you don't see your logo here yet, please get in touch with us if you're interested to join as a member. So quick outlook, what are the next steps of the platform? So we have a strategy paper in a draft status, so we will refine this to have a final draft of the strategy ready soon. Then we will also further specify the platform's topics and map this also with the Austrian expertise and community. Then I can announce also the next board meeting for all platform members and interested parties. It is the 12th of December in Graz and online. So people who can be in Graz will meet at the BNN office and all others can connect online. It will be in the afternoon from 4 to 5. And also in April 2023, sorry, this is a typo, will be the Bio-Nano-Med conference in Graz, and NanoMedicine-Austria will sponsor the Best Talk Award. So I hope there will be many interesting submissions to oral talks. So this was already the end of the introduction and also the information about NanoMedicine-Austria, the platform in general. As said before, it's an open platform and we would be happy to get more interested experts that interact with us. So please feel free to visit our website and get in touch with us and also subscribe to become a listed member. Okay, with this we can already go into the next part of the webinar, which is the presentation of the PHOENIX Open Innovation Testbed and its open call. So as I said in the beginning, the whole webinar is focused on PHOENIX and also the Austrian partners within the PHOENIX project. So what does PHOENIX stand for? PHOENIX stands for the Pharmaceutical Open Innovation Testbed for enabling nano-pharmaceutical innovative products. So what is the specific challenge behind? There was a call in Horizon 2020 that is called Open Innovation Testbeds for Nano-Pharmaceuticals Production. It's an innovation action. And the aim is really to establish this Open Innovation Testbed for nano-pharmaceuticals. And this is needed because many novel promising lab point-of-care nano-pharmaceuticals are more or less waiting in their lab to be translated to become a real product on the market. And nano-pharmaceuticals have a strong potential for providing more effective and safer therapies and diagnostic procedures for a wide range of diseases. But of course, the major challenge to produce the novel nano-pharmaceuticals to GMP quality in sufficient quantity for late preclinical and clinical testing remains. So what is the main prerequisite for a successful implementation? It's definitely affordable and advanced testing, manufacturing facilities and services for novel nanopharmaceuticals. And the difficulties in industrial scale production lay in terms of product quantity and quality required for the entering for clinical trials. So it's really this translation from the preclinical phase through the clinical phases, and then providing all the needed upscaling quality that you can also fill the dossier and get the regulatory approval to get it then on the market. So the objective of PHOENIX is really to enable a nanopharmaceutical innovative product and to bridge this valley of death between science and the nanopharmaceutical product when it enters the clinics. So PHOENIX really aims to enable the seamless, timely and cost-friendly transfer of nanopharmaceuticals from lab bench to clinical trials by providing the necessary advanced, affordable and easily accessible PHOENIX Open Innovation Testbed, so OITB. So who are the partners within PHOENIX? The project consists of 12 partners from different European countries, as you can see here on the map shown. And these partners contribute different complementary expertise to cover all the different aspects that are needed to really translate nanopharmaceuticals to the market and bring them from bench to bedside. So the project budget is around 14.5 million euros, and the requested EU contribution is more than 11 million euros, which is then distributed between the 12 partners. And as said before, the partners have different expertise and they provide also their own resources and services to boost the first implementation phase of the Open Innovation Testbed from the project start. So since more than a year now already, PHOENIX is a running project. And as you can see also in Austria, there are 3 dots. So we are 3 Austrian partners in this 12 partner consortium. So what is the PHOENIX concept and the service strategy? On the left-hand side on the slide, you see the different parts that are covered within PHOENIX. This ranges from product definition, technology development, product development, but also quality management, risk and safety assessment, regulatory support, upscaling, sustainability, training and marketing. So more or less the whole range that could be needed or is needed. And for the services, you see here the different topics or overall topics. So we start with physical chemical characterization, and then we go through in vitro characterization, in vivo characterization, manufacturing, so all services that are needed for the scale up until you reach the GMP manufacturing of a nanopharmaceutical. And then we have also innovation support within the services, so that everything from the innovation phase can be also supported and covered. So for the PHOENIX work plan, you see on the image the different work packages that cover the different topics. For the service portfolio establishment, there are 5 demo cases that are covering 3 delivery routes. So in vitro, in IV, oral skin, and also 1 diagnostic agent. And also in these 5 demo cases within the project, we have 4 nanopharmaceutical types covered. So nanocrystals, lipid vesicles, particle conjugates, and also 1 polymeric diagnostic agent. So this is what is covered within the PHOENIX consortium. And then there is also the service portfolio validation with 2 pro bono demo cases. So there is an open call for any end user of any service, and up to 100% of the costs can be covered by the PHOENIX project. So for this open call, there are services offered in 4 different areas, as said before. So physico-chemical characterization, in vitro characterization, in vivo characterization, and also manufacturing. And any legal entity and research group based in the European Union or associate country can apply. The open call is open now until end of January, so this is the deadline to apply. Then the notice will be given by 1st of March, and the second stage application then has the deadline end of March 2023. And you can find also all relevant info on this link there on the website of the PHOENIX Open Innovation Testbed. So just again that you get an impression of what are the offered funded services in the 4 different areas. So really a broad range of different things are offered there that you can potentially use to boost your nanopharmaceutical idea or technology. And yeah, just again, the deadlines, this is really now the call that you apply until end of January and make use of this open call and of the PHOENIX Open Innovation Testbed. So this is already the end from the overall PHOENIX presentation and about its open call. And now we switch to the presentation of the specific Austrian Partners expertise. So for the first talk in this part, I hand over to Hannes BAUER from RCPE, who will talk about digital process design for nanoparticle manufacturing. So Hannes, the floor is yours.
Okay, so hello to everybody. My name is Hannes BAUER. I work for the Research Center Pharmaceutical Engineering in Graz, Austria. And my talk is about digital process design and the work we do in this area within the PHOENIX project. So at the Research Center Pharmaceutical Engineering, we are a group of 20+ people, and we deal with simulation and modeling. That's a small area in the Research Center for Pharmaceutical Engineering, where we have a number of modeling and simulation tools that we use routinely, and these tools are focused to pharmaceutical process and product design. So for example, we use computational fluid dynamics to study mixing processes, the discrete element method for powder flows and for simulating the flows of tablets, for example, in a coater. We also look at reduced order models that can simulate very fast unit operations and combinations of unit operations. And also molecular mechanics, molecular dynamics is a methodology to predict the behavior of molecules with each other. For some applications with complex fluids, we have a method that we develop ourselves called smooth particle hydrodynamics. This method has some advantages in some cases, but it's not relevant here actually. So how do all of these methodologies fit into PHOENIX? So the idea, and we have heard it before already, is that you have a product at the lab scale, and you somehow want to have enough product in a clinical for clinical trials in GMP quality. So there are several steps that are in between. Technology transfer, scale up, and all of this in a GMP compliant way. And we think that our models can help with that. So the goal within PHOENIX was to take what we already know, our simulation methodologies, and apply them to nanoparticle production processes and create a simulation framework for digital process studies. So we do this for 2 processes. One is nanocrystal formation, which is a top-down milling process, and the other is a nanoemulsion formation, a top-down dispersion process, if you want. So my talk is actually not about these details here, but I want to provide a more general overview of our modeling approach and how this would also maybe fit to other nanoparticle processes. I think that makes a lot of sense here to do that. So if you would look, so we view all of our simulation methods in this quality by design framework. So this quality by design framework is accepted by regulatory agencies now. There are ICH guidelines out there. And the idea is to favor systematic process understanding, right? So the tagline is that you should design your processes for a specific quality instead of testing it after the production. So usually you do this by establishing a link by using by establishing critical material attributes and also finding out where the critical process parameters are. And this would allow you then to predict critical quality attributes. And the simulation models that we want to have work similarly. So these things match. You would have in these simulation models, you would have material attributes and process parameters. And the simulation result would then be the quality attribute, for example, particle size or particle size distribution. So the difference between those two things, I mean, they conform to each other, but the difference is that this quality by design approach is usually a black box approach, whereas if you put a simulation model into this black box, you try to integrate a lot of physical models of mechanisms, and so you can get a lot more out of this model. So if you try to apply this to nanoparticle production processes, you will find that there is a multi-scale hierarchy of phenomena. So at the device scale, this could be a mixer, for example. You would have flow rates, pressures, temperatures, compositions, and also the distribution of temperatures and compositions. At the product scale, the actual nanoparticle, you would have the formation kinetics, aggregation kinetics, the assembly of various molecules, and also breakup and dispersion phenomena. So this would be the mesoscopic scale. And of course, if you break it down further, you end up with molecules. And these actually make up the physicochemical properties of the entire system. So all of those things are, of course, interconnected. But there are tools for each one of those scales, right? So for example, at the molecular scale, we have molecular dynamics. At the device scale, we have computational fluid dynamics, the finite element method, and others. But the idea is, or what you have to know is that all of those methodologies only work at a certain length scale, right? So for molecular dynamics, you can only do up to 10 nanometers. Whereas at the device scale, you can resolve the entire device, but you cannot resolve it fine enough to actually resolve the nanoparticles. So you need a scale in between, and this is this mesoscale that's especially important for nanoparticle production processes. And this would be basically either statistical models or coarse-grained molecular dynamics simulations. So the methods for this are available. It takes time to connect all of these things. And if you do that, you end up with a simulation model that can demonstrate fundamental understanding of these complex systems. Also, because there's some mechanisms built into these models, they can serve as a digital magnifying glass, and you can extract more information out of it. If the model is then validated with experimental data, you can predict target quantities, your critical quality attributes, narrow down the design space, optimize operating conditions, and this ultimately results in reduced uncertainty, reduced risk, and improved quality, and ultimately you can bring the nanopharmaceuticals faster to the patient. So what you need to do though, is you need to do some work beforehand, and when you do that, the model stays actually with you over the entire product life cycle. So it's not lost once you have done it, but you can also maybe adapt it to different devices or use it for scale up to even larger product quantities. So these are the benefits of the simulation model. There are some challenges that we face when we want to produce such a model. For example, we always deal with dispersed multi-phase systems. And that means that if you have a computational cell, so that's the smallest resolution that you the smallest entity that you can resolve in this model, a spatial entity basically. And this entity, this computational cell, is larger than the particle size because nanoparticles are just so small. So it's a challenge to keep track of these nanoparticles in a system that's actually in a computational system that's actually larger than these. Yeah? And also you need to keep track of many of these particles because there are just so many of them. Also, the interactions are increasingly important because these are often dense systems, so they have like 10 volume% and so on. And that's also quite a challenge to address. Further, the next challenge that we face is, for example, turbulence modeling. So if you have these processes and you want to disperse, for example, that these are always high energy processes. So you automatically end up with a turbulent system. And that means that fluid properties vary chaotically in space and time. For example, as you see here in this jet, in this picture of a jet here. So your computational grid, which is shown here way too coarse, needs to actually be resolved a lot more finely. And that ends up, and that amounts to a large number of computational cells that you actually need to solve a flow simulation like this. And also small time steps are required. So this means that the computational requirements for this are on the order of days and not on the order of minutes or hours. The next challenge that we face is that at the mesoscale, so at the actual product particle scale, we don't always have the models that capture the physics. So basically, for example, if you want to do a milling process, the breakage kinetics at the nanoscale are not really known, and the same thing is true for the dispersion kinetics at the nanoscale. So the models are there for larger systems. So this is chemical engineers have tried to solve these challenges for larger systems, but for nano particles, this is still a relatively new topic. The next challenge that we face is that our models require physicochemical properties. So there are physicochemical properties required for the governing equations. And these bulk properties are often uncharacterized, and it's especially true if you have an interface. If you have different phases that mix, the interface properties are, first of all, difficult to characterize and often are just unknown. Also, these properties, they change with the local thermodynamic state. So they change with pressure, temperature, and composition. And if you have a process that that varies those in 3 dimensions and maybe over time as well, you can end up with quite a difficult system. So to summarize, I want to say that tools for integrated process and product design for nanopharmaceutical processes are available. You have to deal with multi-scale phenomena if you want to work in this quality by design framework and you want to actually predict target properties on, for example, particle size distribution. There are nanopharmaceutical specific challenges that remain, but I think it's worthwhile to pursue them because the simulation model that you can generate is valid over the entire life cycle and can be used again. So with this, I'm already at the end. And I'm happy to answer any questions in the following Q&A session. But you can also reach me here on this email address if you are interested and want to hear more of this. Thank you.
Perfect. Thanks a lot, Hannes, for this interesting presentation. As said before, we will have the Q&A session at the end of the webinar. So this is a reminder again for all participants. If you want to ask something, use this questions section and just type your questions there. So thank you, Hannes. And with this, I would like to hand over to the next speaker, who is Paul Gattinger from RECENT. He will talk about infrared and Raman spectroscopy for PAT applications. So Paul, nice to have you. Thank you for your contribution today. I already see you. And we also already now see your slides. So all fine here. So Paul, the floor is yours.
Thank you very much. Thanks for the introduction. I will today give a short overview over infrared and Raman spectroscopy and the possibilities of how to use them in process analytical technology applications. Just before I really start, I want to give a short overview over my company. We are RECENT, stands for Research Center for Non-Destructive Testing, located in Austria. And we were founded in 2009, are around 40 researchers and do funded and non-funded and contract research. The company is basically divided into two areas, an optics and acoustics area. And in the optics area, we are three groups: infrared and Raman spectroscopy, that's my group; optical coherence tomography, terahertz technology. And in the acoustics area, we are also three groups: laser ultrasounds, photoacoustics, and physical and computational acoustics. And now let's dive into the presentation. First of all, I want to clarify what is infrared and Raman spectroscopy. And to answer this, I have to first tell you what infrared radiation is. Infrared radiation is electromagnetic radiation, and if you look at the electromagnetic spectrum, it sits right between visible and terahertz and microwave radiation. Today, I will be focusing on near infrared and mid infrared. The near infrared radiation starts at about 800 nanometers and goes up to 2.5 micron wavelength. And the mid-infrared range is starting from 2.5 and goes up to 10 to 20 micron wavelength. And the remaining part is then usually referred to as far infrared. So what is infrared spectroscopy? In infrared spectroscopy, we measure the molecular absorptions of infrared light. Molecules, they absorb specific parts of the infrared radiation, and the infrared radiation induces molecular vibrations. In the mid-infrared, we are usually dealing with fundamental molecular vibrations, and in the near-infrared, we are usually dealing with overtone and combination vibrations. So just a few examples, or just the types of vibrations that can occur are depicted here. We have the symmetrical stretching and asymmetrical stretching and scissoring, rocking, twisting and wagging vibrations. And just as an example, I brought here the near-infrared absorbance spectrum of water. We can see the water absorbs the infrared light, for example, here at 1900 nanometers roughly, and it induces molecular vibrations. So the molecules absorb and start to vibrate in this fashion that is shown here. For example, at this 1900 nanometer, we have asymmetrical stretching and scissoring vibrations. And for example, here at 1450 nanometers, we have symmetrical stretching and asymmetrical stretching. So now I talked a lot about that the molecules absorb infrared radiation, but how can we actually measure that? And it can be done with so-called infrared spectrometers. And there are basically three different types. Of course, there are a lot more variations of these different types, but more or less, these are the three different types that we have. First one is dispersive spectrometers. This is, I think, probably the most well-known and common one among people. just compare it to visible light going through a prism, and you will see a rainbow. And this is more or less an analog phenomenon, but just in the infrared. So we have infrared radiation, send it through a prism, and then we have the spectrum in the infrared, and we can measure this spectrum with array detectors. So every pixel in this array detector detects a different part or a different wavelength, so to say. And that's how we get our infrared spectrum. Then we have tunable Fabry-Perot filter spectrometers. They work a little bit differently. We have broadband infrared radiation entering optical resonator. These are two semi-transparent mirrors, and depending on the distance between those mirrors, only a specific wavelength can transmit this optical through this optical resonator. And by tuning the distance between the two mirrors, we can again record a spectrum here with a single point detector. And the last the last type is the Fourier transform interferometer. This is more or less the gold standard in infrared spectroscopy, where the infrared radiation enters an interferometer and we measure an interferogram. And by applying a Fourier transform on the interferogram, we can retrieve the infrared spectrum in the frequency or wavelength space. Then I want to explain to you what Raman spectroscopy is all about. It uses the Raman scattering effect, which is an inelastic scattering effect. This Raman scattering is usually induced by a monochromatic light source, so it's mostly red or green lasers. And the Raman scattered light is shifted towards lower frequencies usually, but sometimes also towards higher frequencies. But in the way we use it, we detect the lower frequencies. And this shift corresponds to wavelengths or frequencies of fundamental molecular vibrations. So what's the difference to infrared spectroscopy? The shifted wavelengths is still in the visible or near infrared regime. Which means we can use silicon detectors to measure it. And this is, for example, every mobile phone camera uses such kind of detectors. So it's very abundant and easy to use detectors. Then the infrared and Raman spectroscopy are complementary techniques, which means that Raman spectroscopy is sensitive to homonuclear bonds, for example, C bonds or oxygen-oxygen bonds. And infrared spectroscopy is sensitive to polar bonds, for example, CH, so carbon-hydrogen bonds, or hydrogen-oxygen bonds. And that's the difference between infrared and Raman spectroscopy. Now I want to tell a little bit about the application possibilities of infrared and Raman spectroscopy in process analytical technology. Near infrared is already a well-established technique in process analytics technology. Many different materials and chemicals can be measured in the process, and effective chemical analysis can be done by multivariate data analysis. And we call that chemometrics. And usually the measurements are real time, and very often they are even contactless. And the near infrared and Raman spectroscopy are highly compatible with fiber optics, which also makes them ideal for in-process applications. In the recent years, we have experienced interesting technological advancements, namely the miniaturization of spectrometers. As you can see here, modern filter spectrometers are, for the near infrared, are quite small. they usually fit just right in the palm of your hand. And before, such spectrometers were like 19-inch racks or whatever, and now they are pretty small already, which makes them also cheaper, of course, very rugged, and very interesting for many applications. And speaking of applications, I want to show you 3 different application examples of process analytical applications. For example, here we measured formaldehyde concentration in the formalin production. This was also done with a microspectrometer and an optical fiber and an immersion probe. And we did calibration and validation measurements, and it looked quite well. Long-term measurements of this reaction showed that the inline measurements shown in blue here correspond well with the offline reference measurements, and the company could estimatedly save 200K per year, and the working conditions have improved, of course, because there's less exposure to hazardous materials for the workers, and also many more benefits, of course, because the resources can be used for different things now. Another example is moisture monitoring using near-infrared spectroscopy, especially now that energy prices are high and climate change is also a thing. Many industries are interested in not over drying their products or just monitoring the moisture in order to stop the drying process at the right time. And we did that, for example, in the food industry for dumpling bread and starch derived products. And as we can see here as well in these graphs, near-infrared contact lens near-infrared spectroscopy with that it's possible to track the degree of drying very well, and these things will be used a lot more even in the future. And we are actually also in the scope of the PHOENIX project trying to measure the drying process of nanoparticles as well. Another application example, but this time for Raman measurements, is we measured the in situ mild steel corrosion. We had a steel sample in an artificial corrosion chamber over the course of 8 days, where salt and fog cycles were made, and we could see the chemical transitions during the corrosion cycle, which is depicted here. You can see just in different salt, fog, and drying phases, different molecules that emerged or different corrosion products. And this is very interesting because it shows that Raman spectroscopy can be used in very hazardous environments. So in this case, it was a salt fog environment, which is for measurement devices usually not such a nice environment. Yeah, and I'm already at the end now of my presentation. I just want to sum up what I talked about. So basically, I explained briefly what infrared Raman spectroscopy is all about, and that it's a non-destructive method for chemical analysis. And we have basically 3 different spectrometer types. Contactless, but also in contact real-time measurements are possible for PET applications, and miniaturized spectrometers allow even more applications, and they lead to strong cost reduction of such systems, which makes it more interesting also for smaller companies. And I want to thank the infrared Raman group at RECENT, and also our external partners and funding agencies as well. Thank you very much.
Perfect. Thanks a lot, Paul, also for this presentation. Again, the reminder to the participants that if you have any questions, you can type them in the questions area. So now we can change to the last presentation of the Austrian PHOENIX Partners. So after RCPE and RECENT, it's again me for BNN, and I will talk about safe and sustainable by design and its added value for nanopharmaceuticals. So what is safe by design? There is someone talking, sorry. So safe by design is a concept that is meant to integrate safety considerations already in the early innovation phase. So usually any type of technological innovation presents a challenge to health and environmental risk assessment. This is also valid, of course, for nanopharmaceutical innovations or pharmaceutical innovations in general. And the rapid innovation causes a gap between technology and suitable risk assessment tools and frameworks. And to reduce uncertainties, but also minimize all kinds of risks to humans and the environment, which is not addressed totally yet in the pharma sector. Starting at the early innovation phase and cover the whole innovation chain is called the safe by design concept. So this is really a concept that was established in the last years coming from the nano safety perspective. What is the benefit and the added value of safe by design? It's definitely the timely and early experts interaction. So in this picture, you see the ideal situation in safe by design is the very early interaction and communication between the innovator and the safety expert, and that different things are addressed like material, process and product safety, but also regulatory and ethical aspects and public perception. And in the best case, this fosters the innovation process and makes things going faster to the market. So instead of doing all the risk assessment at the end of the innovation phase, you do it already at low TRL and make everything safer and optimize things from the very beginning. Of course, in the pharma sector, human safety is one of the key things when you develop something new. I mean, this is addressed very early, also cytotoxicity, etc. But this includes also the whole environmental part, so environmental safety topics. Recently, this safe by design concept was extended towards including also sustainability aspects. So just a quick reminder, sustainability, of course, you have the 3 dimensions addressed, so planet, people, and profit. So it's really including all societal, environmental, and economic aspects. And it is a very broad topic that for sure includes safety, but also goes far beyond. So a sustainable material must be safe, but a safe material is not automatically also sustainable in all different aspects. So to include sustainability into the safe by design concept, it was called safe and sustainable by design concept. Just to make you aware that SSBD is also strongly addressed in different policy and related documents. So definitely sustainability is a cross-sectoral hot topic, and it has a high priority in the European Union, the UN, and also their strategies. I listed here a couple of relevant documents like the 2030 Agenda for Sustainable Development, the European Green Deal, but also the EU's Chemical Strategy for Sustainability. So this is now not pharma specific, but for instance, in the Chemical Strategy for Sustainability, there is also a concept announced that is called One Substance, One Assessment. So this means that for chemicals, you don't have to do the assessments in all different applications, but for the chemical ones. And this would then also influence potentially the pharma sector. So for SPD, the key pillars, there was also a framework developed by the Joint Research Center from the European Commission. So two important documents were published recently. The one is called Safe and Sustainable by Design Chemicals and Materials, Review of Safety and Sustainability Dimensions, Aspects, Methods, Indicators and Tools. And the other one is really about the framework for the definition of criteria and evaluation. And in these documents, it says that safe and sustainable by design is an approach to the design, development, and use of chemicals and materials that focuses on providing a function or service while reducing harmful impacts to human health and the environment. And this JRC framework is structured as you can see here. So in the design or redesign phase, there are 8 different SSBD principles that range from material efficiency, minimize the use of hazardous chemicals, materials, but also design for energy efficiency, use of renewable sources, prevent and avoid hazardous emissions, reduce exposure to hazardous substances, design for end of life, and also consider the whole life cycle. So this is based on different principles like green chemistry, green engineering, sustainable chemistry, and safe by design. And then in the sustainability assessment, you see different steps. The first 3 are covering the whole safety aspect. And then step 4 is about environmental sustainable assessment, so life cycle assessment. And then the step 5 about social and economic sustainability assessment. And this should cover also the whole life cycle from starting with the material, then how it is used and produced, its end of life steps, and also potential recycling. So what is the added value of SSBD? It's really that different settings and options during the innovation process are assessed with the clear aim to design materials, processes, and also interim or final products as safe and sustainable as possible, while keeping the technical requirements and complying with current and future regulations, and also keeping costs as low as possible. So now the link to the pharma sector. So the specific challenges in pharma. I wanted to do the story around COVID because with COVID-19, the pandemic, it was shown that pharmaceutical sector in general has a great ability to respond to urgent circumstances and requirements. So this was addressed fast, but nevertheless also we still need to think that we need to improve towards the sustainability goals. So it was shown with COVID that rapid developments can be done by pharma if needed and if there is a high demand, but still the environmental impact also needs to be considered and addressed. So there is this concept of pharmaceuticals route to the environment. So as you know, pharmaceuticals are produced by industrial facilities and are consumed in agricultural production, hospitals, healthcare facilities, and households. And during their use and production, they are released into the environment through different routes, for instance, urine or wastewater. And in Europe, there are 88% pharmaceutical release into environment from patients' use, and 10% more or less come from incorrect disposal of products, and 2% from production. Another reason why pharma sector is in the spotlight is because it's a major contributor to global greenhouse gas emissions, and the impact on the environment is multifaceted. So the impact derives from R&D activity, supply chain, transport and logistics, disposal of pharmaceutical products, and also the chemistry. And a study from 2019 found out that the pharmaceutical industry emission intensity is about 55% higher than that of the automotive industry. I already talked a little bit about policy context, just to let you know that also FPA, the European Federation of Pharmaceutical Industries and Associations, is addressing this topic. So they really try to also build a healthier and more environmentally sustainable future. So the pharmaceutical industry is committed to this goal, and they also support the overarching objectives of the chemical strategy for sustainability towards a toxic-free environment. So SFPD can definitely support to address this challenge of pharmaceuticals released to the environment, and consider this also already in the innovation phase, in the low TRL level. So as I said, sustainability is to become a high priority in the pharma sector. SFPD supports to find the best solution from an early innovation phase. And definitely a continuous optimization is needed to become more sustainable. And of course, the higher the TRL, the more data should also be available. And then to claim and prove that SSPD is embedded in your innovation pathway within your company or institution is a clear benefit compared to your competitors, and also makes your materials, process and products future-proof. But since it's a very broad topic, for sure, multidisciplinary experts cooperation is needed to properly address this. And what we try from BNN is to guide and support you towards implementing SSPD, and also to keep pace with latest developments in this area. So with this, we are finished with the presentations of the Austrian partners of the PHOENIX Open Innovation Testbed and Project. So we enter now into the Q&A session. So there is a question to Hannes that is called, what is the number of particles they can simulate? What kind of computers do you use? And do you have access to the scientific clusters?
So what we here use, we use mostly GPUs, yeah, graphic cards. The number of particles that we can simulate ranges from millions to tens of millions and even more. So it depends on the exact hardware that we use, but with the like advancement of those, right? So these GPUs always try to get better and better. We can simulate each year like twice the amount of particles than the previous year. Yeah? So it's really tens of millions, like 10 millions or so on. There's no problem at all.
Okay, thank you very much for answering, Hannes. There are no other written questions. Maybe any of the platform chairs do have a question that they want to pose now to the presenters? If so, just feel free to ask your question now. Otherwise, I think all of us also edit contact details into the presentations. So maybe there is some follow-up then via email with the presenters bilaterally. Okay, so with this, I would also like to quickly announce that there is Austria-Bulgaria bilateral call. So for scientific and technological cooperation from ÖAD. So the closing date for this would be end of January. So the 31st of January at noontime. And as copied in here, funding will be provided for the mobility of researchers to carry out bilateral research projects. And the project duration is from 1st of July 2023 until 30th of June 2025. So if somebody is interested to apply for this Austria-Bulgaria call, you can also get in touch with us. And in the meanwhile, there is another question that is, we do have some participants also from the Medical University of Sofia, which is perfectly fitting to what I just presented about this call that is open. And the question is, what are the possibilities for collaboration with you? Okay, yes. So thank you very much for this question. Do you also want to quickly introduce yourself and your expertise? Then I would like to ask you to raise your hand so that we can give you the right to talk.
Hello,
Great.
can you hear me?
Yes, we hear you now.
Okay, thanks for the invitation to participate to this event. In fact, it was my friend Thomas Moore, who is one of the chairs of your action, who invited me. Well, our group is working on nanotoxicology and all kind of preclinical toxicological studies. We are part of the Faculty of Pharmacy in the Medical University of Sofia, and we work really tightly with a group which is doing pharmaceutical formulation and drug delivery systems in the nanoscale. So I think there are topics that could be interesting, and we could discuss regarding research. So my question was, are you more focused towards manufacturing, or also there are these possibilities for purely research activities that we could benefit together from?
No, definitely. So in the platform, we definitely have many researchers that are doing things that are more related to research than to production or manufacturing. So for sure, there will be some interesting partners that could collaborate with you on this topic. But of course, we need to connect you with them, and then you can work on the idea specifically. But I would be happy to facilitate this collaboration and bring you in contact with people after this webinar.
Okay, great. So maybe I could discuss with all of the groups that we work together and think about some of the connections that would be best to form.
Yes.
So that's a great idea.
Perfect, perfect. Excellent. Thank you very much for this contribution and also for joining our webinar today. It would be nice to see a real application then for this bilateral call end of January.
Yep, totally. Thanks.
Perfect. Thank you very much. Okay. With this, we already reached the end of the webinar. So just to remind you again for important dates. So the board meeting that will be in 2 weeks as a hybrid event in Graz and online in the afternoon. And again, the call to contribute also to the BioNanoMeet conference taking place in April in Graz, where we will sponsor the Best Talk Award. And it also fits perfectly that BNN is in the gift-giving spirit, as it says here. So if you want to have a pre-Christmas gift, you can try your luck and contribute to our raffle that is currently open. So if you subscribe to our BNN Quarterly, which is our newsletter that is released 4 times a year, you are registered automatically to a raffle that is open until the 12th of December, so 2 more weeks. And if you are lucky, you can win participation to a conference, which can be either BioNanoMeet or the NanoSafe. So it's up to the winner to choose which conference is more interesting. So yeah, try your luck and maybe you can get yourself a pre-Christmas gift. And with this, we are at the end of the webinar. I would like to thank you all very much for your participation. Big thanks, of course, to the chairs of the NanoMedicine-Austria platform that are always committed and yeah, really contribute to make this platform a success. And of course, a big thanks to the speakers, to Hannes and Paul, for presenting also the Austrian PHOENIX partners in today's webinar. As said in the beginning, the recordings will be made available, so you can rewatch. And yeah, please keep in touch with us and happy to follow up. Have a good day. Bye, everyone.